We're continuing, as you see, with a lot of bio. And part of the reason why there's a lot of bio is not because everything we do at the Nano Center is bio. It's just because it happens to be a topic of a lot of interest. And Beck-Barkay is the next speaker. He's going to talk about Nano Biophysics. And this is a very, very neat combination that you've seen in previous presentations, but Roy will emphasize, I think, some new and exciting issues.
All right. So, when Yael told me that we are going to have this popular science talk, I had a different thing in mind than what the previous speaker had. And today I'm going to talk about nanobiophysics, and I don't know how much we'll be able to decode what is nature mysteries. But I want to give you my perspective about what is the title, what is nanobiophysics. But before I'm going to do that, I'm going to start with a joke. So this is always a good thing to start so the audiences are awake. So the joke goes like that. About the relationship between physicists, theoretical physicists, and milk production. So I'm a physicist, and I'm a physicist by training, and I got into the business of biophysics pretty much in the last 6 years or so. So it goes like that. So the milk productions in a dairy farm was very low, so the farmer wrote to the local university asking for help from the academia. Okay, so this is what they asked Fernando and Danny and Shahar and all of them. They ask help, right? So they had a real serious problems with milk production. So the economy professor said, okay, I just need $10 million. I can make up design a nationwide campaign that will increase the profit by 5%. We will not do anything to the milk, but it will increase the profit so the farmer will be happy. The next letter that they got is from the biologist. Danny wrote them an answer and telling them, okay, I just need $1 million, but then I will be able to design a drug, a special drug. It will be targeted exactly to the site. It will increase the production of 20%, but only for, it will increase 10% of the production, but only for 20% that it will work on. And then they got a letter from theoretical physicists, and he said, I don't need a lot of money. Just give me $10,000, and I will increase the production 5-fold. I said, okay, come on in. We want to listen to you. We don't have any farmers. We don't have a lot of money. What can we do about it? And he said, yeah, I have this great concept in mind, but so far it's only working in the case of spherical cow, radius R, absolute zero temperature and in vacuum. And even the tail needs to go to zero. So that's pretty much the task that the biophysicist is dealing with, and particularly if he wants to have tenure in eventually, that in somehow he needs to design tools to study with physical tools to study the world, but then we are very limited in that sense that we cannot remember all of these leukocytes and all of these the names that were just before. We are thinking in a very simplified world. So the task is how we can not design a spherical cow and still do some good help in biology. So what is nanobiophysics? So the first thing is very small, right? Nano is very small. So nano, we all know about it, that it's very, 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 very small. I just did a calculation a few days ago to find out what is, just in perspective for mankind, what is really nano. So nano, if you think about it, is taking an eye exam, but sitting on the moon. Okay, I'm holding up something here, and I'm taking an eye exam, and I can read what it is while sitting on the moon. This is very far away. This is very tiny objects that we are looking on. And what is biophysics? Now, when I don't know something, so the first thing everybody does, it goes to Wikipedia and looks what is biophysics. And if you look on biophysics, so it's an interdisciplinary science. Okay, I'm in the right center. I'm in the nano center. We are all interdisciplinary. Chemistry, physics, engineering, everything is there. That study biological system, okay, that I can understand. But then it goes to, under the branches spans all level of biological organization, from the molecular scale up until ecosystems, and doing that with physical science methodology. That's pretty much everything any biology done in 2012 is doing. Okay, everybody is using physical science methodology in order to study biology. So that's one occasion that Wikipedia might not be so correct. So what is biophysics in my view? So biophysics, at least in my view, is trying to study the key physical interactions. After all, even in biology, the interactions between molecules or interactions between cells are governed by physical laws, even if they are very, very, very complicated. And the idea of a biophysicist is to actually study those interactions, trying to find out the formulation that summarize those interactions in biological systems. And the building blocks that we have are very, very complicated. First of all, we have a lot of them. As a physicist, when I was a solid-state physicist, if we had a system with 4 atoms, that was so difficult that we couldn't understand how to calculate correctly. We had to do a lot of assumption. In biology, it's so much more complicated because we have proteins, that each protein is made out of 20 different building blocks of the amino acid. We have fat with gazillion numbers of different types of molecules. This is by the way, the diet that you need to eat, right? So you have carbs, all the sugars, and you have the nucleic acids, the genetic material that is also made out of 4 different building blocks. So we have tons of building blocks. Each one of these building blocks is relatively small. It's a molecule. It's a molecule. It's even smaller than a nanometer. So if you want to think about what is nanoscale biology, you immediately understand that nanoscale biology is not about a single molecule. Is, as you saw before in the previous lectures, it's all about complexes. Okay? And complexes is what made up naturally in your body. And all these complexes are actually on the nanometric scale. This can be the skeleton that is inside the cell that I'm studying very intensively. It can be like the lipid and nucleic acid like Danny is doing to make complexes that will be delivered as drug delivery. It can be complexes of different, you see here it's fat with protein. This is fat with nucleic acid. And it can be even like tiny objects like viruses that are exactly on the nanometric scale, and the materials that makes them is protein and nucleic acid. So that's pretty much what is nanobiophysics is all about. And the key problems that we have is that it goes back to the third slide or so that I showed you. The key problem is that these complexes are very, very small. We need to somehow look on them, and it's very difficult. We cannot do that with regular microscopy. This is too small for us. And the other problem is that because I'm a biophysicist and I'm interested in the interactions between them, I need to be very careful that anything that I'm doing in order to measure those interactions will not be disturbed by the markers that I'm putting in, because those markers are themselves on the nanometric scale. So I have an inherent problem that I'm trying to measure something, I'm putting something on the same length scale, and then I'm trying to understand the interactions that are again on the same length scale. So I need a very, very different kind of techniques that I will be able to look on something so small without disturbing it. So how can we study nanoscale biological complexes? The first thing that you need to do is that you need to have an extremely good and talented group. And that's what I have. And in fact, I'm very, very honored to have such a good group in the Nano Center where I'm working in. And the other thing that is very important for you, that it will be interdisciplinary. If you want to do biophysics, you must not just say it in order to get grants or so. You need to actually do it in practice. So I have students from physics, from medical school, and from chemistry that are actually doing the research. Right? We are not doing actually anything anymore. We are just, telling them what to do. So, but they are actually doing all this job, and they are coming from the different disciplines, and their education is actually extremely relevant to what they need to do. So this is the students around Micha, Guy, Eti, and Rona, and my assistant and lab manager, which is Adi, and she missed. Okay. The second thing that you need to do, biophysics, is that you actually need to have a lot of money. And you need to have a very, very expensive setup that will be able to give you this resolution, this nanoscale resolution. And this cutting-edge technology that we are using in our lab is called small-angle X-ray scattering. This is, in fact, the system that we build in my lab. And it's composed of you see the price tags for the components in it. And that was built last year in Tel Aviv here. And the idea about this technique that is actually you can shine an X-ray, very, very directed X-ray ray into a sample which contains these complexes, and you can keep them in this watery environment. You don't need to dry them up. You don't need to put them in vacuum. The radiuses might be of an R, but you need to keep them in the right environment because you are interested in the physical forces and interactions that are there. The second thing that is very important, you don't need to tag them with anything. You don't need fluorophores, you don't need heavy metals, you don't need anything. You just need to put it in the X-ray, and you get the structures that is relevant for you, and the structures that we can measure is in the range of 1 to 100 nanometers. So this is why we fit to the Nano Center very well. So this is a little bit about the projects that we are working with. So these images I just got this morning from Ram. So I was able to put them in the presentation in the end. And Ram is working on a very exciting project. This is one of the projects he's working on. This is actually to study the structure of things that are not structured, of things that have a lot of flexibility inside of them, proteins that they don't fold into a specific configuration, but sample a lot of different configurations while they are in water. And this is done using advanced methodology and computer science and aids of computers in order to take a what seems to be very boring signal into these very exciting cartoons that tells us a lot about the functionality this protein have. A different project is Guy that was asking Danny before a question and actually is working with Danny. So he was a spy. And he's actually working on this lipid vehicles to carry drugs. And the idea that we try to understand what are the physical mechanisms that hold these nanosubmarines in place, and what can we do about these interactions that holds it and breaks them apart. What can be mechanism, what treatment we can do after we understand what are the physical forces that holds them together. So again, this is another excited research area. The third one that I'm going to show you is done by Eti and Micha. Eti and Micha are actually looking on the cytoskeleton. The cytoskeleton is actually the if you pop up a cell, you won't get just water, you get jello inside. And this jello is made out of filaments, and this filament is called a cytoskeleton. So they are actually studying those cytoskeleton using X-ray diffraction to understand what are the mechanisms that actually holds this skeleton of the cell in place. So that's pretty much it. So that was a really quick and a general overview about what is biophysics. And the main reason we are doing it is it's not really, we are not helping so many people as you, Fernando. We are taking our hat off. We are just doing it because it's so much fun. That's it.